Yeah. It's working, Hella? Yeah, I think it's working. Okay, great. Good afternoon, everybody. We are sorry for the delay. My name is Jood Hashem. I got my undergraduate degree in Germany, and then I went ahead and got my master's and PhD in the United States of America in Texas. I was thinking thoroughly about what I should be talking to you about today. And I had a lot of things actually to talk about. But then I thought maybe the best thing to do is to walk you through one of my journeys through my research that I have done and my discovery and my enlightenment with calcium. So as you all may know, calcium is involved in several body functions, right? Calcium is involved in muscular contraction, it's involved in neurotransmission, it's involved in bone building, it's also involved in blood clotting and blood coagulation. It's also involved in several cellular mechanisms, such as, for example, cell division, cytokinesis, and also cell signaling, right? By cell signaling, I mean how cells talk to each other, how they communicate inside the body. So for example, one of the very favorite topics of mine that I teach in university is hormone signaling, right? How cells release hormones. And one of the processes that happens is the cells engulfs the hormone in secretory vesicles, right? I don't think I can point to it right here. But what they do is they engulf them in secretory vesicles, as you see here. And only when calcium is increased in the cell, a process called exocytosis happens, in which these vesicles bind to the extracellular surface and release their material to the outside, some of which are hormones, the signaling materials, right? My fascination with exocytosis started when I did my first internship in the United States, right? And what happened is we were learning about it. And it turns out that these vesicles I was telling you about, they have certain proteins on the cell surface, which we were interested about at the time, right? These proteins are called vascular ATPases. So these vascular ATPases, they were in these secretory vesicles, and once they bound or once they merge with the extracellular surface, they are situated at the extracellular surface. You may ask, why is this important? And how does that have to do with calcium, right? Well, it turned out at the time we were interested about vascular ATPases because they were involved in several functions, one of which is angiogenesis. Does any of you know what angiogenesis means? Yes. It's the formation of blood vessels, right? And formation of blood vessels is very important, and it was a very important topic, especially for us when we were looking at diabetic cells and non-diabetic cells. As you all may know, diabetic people are in risk of lack of angiogenesis, right? And this is a very prolonged topic. I won't go there in details. But what I wanted you to know is that we found that there's a difference between diabetic endothelial cells and non-diabetic endothelial cells by the situation of vascular ATPases at the surface, right? So normal endothelial cells have the VATPases at the surface, which at the time we understood was involved in angiogenesis. And the diabetic cells did not have VATPases at the surface, which induced the lack of angiogenesis, right? The question was at the time, then what's the difference between both? Why did these diabetic cells have VATPases on this, no ATPases on the surface, and the normal cells have the VATPases on the surface, right? And then we thought about the next thing, exocytosis. These VATPases are proteins, as I told you, found in these vesicles, and what they do is they merge with the extracellular surface in order to be situated there. We found out that the difference between both is the regulation of calcium, right? The diabetic cells had poor regulation of calcium inside the cell, which prevented the exocytosis process. and those normal cells had perfect calcium regulation, had very high calcium, adequate levels of calcium, which induced exocytosis, which led to the formation of VHPases at the surface. Your next question might be, but this presentation was about cancer. Nobody said we're talking about diabetes, didn't we, right? Our next thought was, angiogenesis is a very important process, correct? And one of the most important cells that need angiogenesis are cancer cells, right? Why? Because they need the nutrients, they need the oxygen, and all of that. And then we thought to ourselves, since the VHPases are helping the vascular endothelial cells to branch out and move out, right? Then what could they do with cancer cells? So we went ahead and looked at breast cancer cells. Ones that are metastatic. Do you know what metastatic means? Metastatic means they move from one place to another, right? And ones that are non-metastatic. And we wanted to see the VHPases on their surface, right? As you may see here, the metastatic breast cancer cells, they express the VHPases on the surface, which later on we understood helped them move and metastasize. And the non-metastatic breast cancer cells did not express any VHPase on the surface. I'm not sure if that's clear from where you're sitting. Normally we dim down the lights so people can see the fluorescence, right? But what you see on the surface is labeled as VHPase. So we saw it on the metastatic cells, but there was none on the non-metastatic cells. Now from our background about diabetic and non-diabetic cells, what was the first thought? Calcium, right? Let's check out calcium regulation in these cells. What if there was no adequate calcium that's helping in the process of exocytosis, right? This is exactly what we did. So we went ahead and looked at the metastatic breast cancer cells, and it turned out they have high levels of calcium inside the cell, which allowed the exocytosis, which in turn allowed for the situation of the VHPase on the cell surface. And then when we looked at the non-metastatic breast cancer cell, as you see, there is very small calcium. Very little amount of calcium inside, right? Which prevented probably exocytosis or downregulated it. And so we don't have enough VHPases on the surface. Now that was very interesting to us, right? But to me, this wasn't enough. I went ahead and looked at calcium and what it does to a cell, a cancer cell in particular. It turned out, it's not working. Okay, I think I'll just improvise. So we have a problem with our PowerPoint. It's not working, but I'll continue the presentation until we get to a point where I have to show you something on the PowerPoint, and then we're stuck, okay? So let me continue the story. So at the time where I found that there is differences in calcium, I thought I wanted to study more cancer cells, right? But before I go ahead and study more cancer cells, I needed to understand what calcium does in cancer, right? It turned out that calcium has several functions in cancer, some that we know about and some that we don't know about yet, right? One of the main processes that calcium is involved in is cellular division. Remember when I said that in the beginning? And one of the most important hallmarks of cancer is cell division, right? Cancer is defined as the uncontrolled proliferation of cells. And so we looked at different cancer cells and we looked at the calcium in there, and we found out that the more proliferative the cells, the higher the calcium is in those cells when stimulated, okay? The lower the proliferation, the lower the calcium in these cells. And then we looked at different processes. For example, calcium is also involved in angiogenesis, as I have previously mentioned, and it's also involved in something called cancer metastasis, which I just have mentioned, right? It turns out that when I looked into cancer metastasis, the regular definition, the movement of cells from the place of, disease to other places in the body, right? So for example, if a person had prostate cancer, and the cancer moved to the bones and then to the brain and whatnot, that is called metastasis, right? But then metastasis, yes.
هلا انت بتحكي انه مرضى السكري مع المعلومه فممكن يكونوا اقل عرضه لاصابه السرطان.
Can we answer questions afterwards? I'll remember your question, okay? It turns out that metastasis is more than just the movement. Metastasis is divided into two main categories: cell movement and cell invasion, right? So we have the movement part where the cells move, and then we have the part where they invade. So they digest the tissue and they invade another tissue, right? So these two mechanisms. And that got just very interesting. So there were many studies that talked about, can I? Okay. Perfect, it's working. Okay. So there are many studies that talked about the involvement of calcium in certain proteins called metalloproteases, right? And these metalloproteases were involved in cancer invasion. And these turned to be calcium dependent, right? So without calcium, they don't work. With calcium, they work. So we were next involved in the other mechanism. This is to show you how cancer is involved in movement and then in invasion. So then we were involved in the next step, right, which is the cell movement. Cell movement was so fascinating, especially to me, when I first put the cells under the microscope, put a camera on there, left them in an incubator, and came the next day. And this is what I saw. The cells were moving. If I were to put normal cells in there, I would not see them moving. But those metastatic cancer cells, they were moving, and they were moving like crazy, right? And so that was fascinating to me, and I wanted to understand, how do these cells move? Because if I understood how these cells move, in my little mind, I was thinking maybe I can stop them, right? And so I went in and I looked at the cell biology book, the one we were taught at undergraduate school, some of us at high school, and I looked, and then I thought, how does the cell move? So it turns out in order for a cell to move, it has to have three main steps, right? So they have sensors, or what we call in science focal adhesions for these cells. So what they do is they put them out, they sense if it's a good place to go, and once they do, they hop in, and so they stick, right? But then their posterior part is still left up. So what they do is they pull themselves, they detach and pull, right? So three steps. They attach, they adhere, they detach from the back and then pull, right? And that's how they move. Three main steps. The funny part is when I looked at these focal adhesions and these proteins that are working, it turns out that, guess what? They're regulated by calcium. Believe it. Yes, they're regulated by calcium. So what happens is, in order for these focal adhesion to attach, they need less calcium. And in order for it to detach in the back and contract, it needs more calcium. So you can see we need less calcium in the front, but more calcium in the back. Now looking at what I know already, remember those peaks that I showed you when we stimulate the cells? We've only seen high stimulation or low stimulation of calcium. To me, this didn't make sense, right? So we have high stimulation of calcium, low stimulation of calcium, then what's with the wave that we have inside the cell, right? Low in the front, more at the back, right? So I went ahead and I looked further into it. And then I went to see how is calcium regulated inside a cell in order for me to understand what's going on, right? It turns out there are different regulatory proteins for calcium. There are proteins that are called channels. You know what channels are? There are proteins at the cell surface that let calcium in, right? There were transporters that took calcium with them as they went in. And then there were pumps that pumped calcium outside the concentration gradient, right? But to me, I was mainly involved, or I was mainly interested in one of the main regulators of calcium. That's what they said, it's a main regulator called store-operated calcium entry. And store-operated calcium entry worked in a very funny way. So this is the cell membrane, right? Once the cell is stimulated, what it does is it stimulates the release of calcium into the cytosol. So we have an increase in calcium, right? Inside the ER, we have a sensor. That sensor senses that some calcium is lost. So what it does, it binds to a channel at the cell surface called ori, or what we call store-operated calcium entry channel, and induces the entry of calcium into the cell. That's why we saw the peak, right? So every time the cell moved, we saw a peak. Yes? Yes. When the cell is stimulated, in terms of a signal, in terms of another cell communicating with that cell, right? Some cells communicate with ATP, believe it or not, right? What happens is it induces the release of calcium from the ER, right? So I have a lot of calcium in the cytosol. There's a sensor in the ER. It senses that there is less calcium. So it goes ahead and binds to the channel to induce it to let calcium back into the cell. So we have an increase in calcium in the cytosol, right? This is how the cell it gets in calcium through store-operated calcium entry. Okay. At the time, I was interested in looking at prostate cancer because prostate cancer at the time had a huge issue over there, and it was the leading cause of cancer death in men. 1 in 6 are diagnosed. But what's important is at the time we had a really good cancer model, prostate cancer model, right? We had a good metastatic cell and a good non-metastatic cell that we knew doesn't work, and we also had normal prostate cancer cells, right? So we went ahead and looked at these prostate cancer cells, and the main hypothesis at the time was these metastatic cancer cells have a lot of that store-operated calcium entry channel, and that's why it's letting calcium in, right? But then I wanted to look at something else. Remember when I told you there's a wave inside the cell? There's less at the front and there is more at the back, right? So first of all, what I wanted to do is I wanted to characterize my cell. So I needed to make sure that definitely my metastatic cell is really highly migratory and highly invasive, and my non-metastatic cell or poorly metastatic cell is poorly migratory and poorly invasive, right? In order for us to do that, we have a method called wound. I'm just going to go here to the video. Is the video working? Lisa? Okay. I think too late. مش راح يزبط. But the video is not working, unfortunately. I would have loved to show you how these cells move. But what's happening is, what I do is I let the cells grow in a monolayer, right? And then I create a wound. So that wound gives space for these cells to walk towards each other, right? They communicate and they walk towards each other. The faster this wound closes, it means that the faster the cells are. The faster the cells are means the more migratory they are, which gives us an indication of how metastatic they are, right? So it turns out that here you can see the wound at when we first did it, and then at 10 hours, and then at 22 hours, the wound was fully closed. It is? ثابت بين 10 Can you press play? Okay, there we go. Okay, so that's the wound, right? I did what I did, as I told you earlier. I created the wound, I put the cells in the incubator, I put a live camera right there, and I recorded for 22 hours. Do you see how the cells are moving? Please notice the focal adhesions attaching, detaching, and pulling themselves. So attach, detach, pull yourself. And that's how they're all walking, right? Now these are metastatic cancer cells. The non-metastatic cancer cells did not move at all. Or if they moved a little bit. But it took around 48 hours, if not more, to see that small movement we're talking about. 48 hours, 2 days. Right? So looking at these cells, we confirmed that these cells are more metastatic. We looked at 4 different cell lines, right? These 4 different cell lines gave us the results. I'm not going to go in details, but a cell called D145 is a very metastatic prostate cancer cell. And the least metastatic was LNCAP, the poorly metastatic. It wasn't moving much, right? Now the next step is to see if they're invading as well, right? So what we do normally is we plate the cells in a well, and on the well we put an extracellular matrix kind of substance. So if the cells were invasive, what they would do is they would ingest this extracellular matrix substance, and they would invade through a pore, a very small pore, and it would go to the underside of the well, right? What we do later on is we label the underside of the well, and we see how many cells were successful enough to invade that extracellular matrix and end up in the other side of the well, right? It turns out, as we saw through the other experiments, the D145 were the most invasive, and the poorly migratory LNCAP were the poorest of all the cells in invasiveness, right? So now that we established that we have these cells that are highly migratory, highly invasive, and then we have cells that are poorly migratory and poorly invasive, our next step was we wanted to see how do they regulate the calcium in these cells, right? Or how calcium is regulated. So these are the cells we want. So the experimental design was we labeled these cells with a calcium indicator, right? And once we induce or we excite this calcium indicator, it would fluoresce. So the more calcium there is in the cell, the more fluorescence, the higher the signal, right? In the highly metastatic cells, as we expected, we saw a very high signal. And in the poorly metastatic cells, we saw no signal at all. But again, that did not explain the wave I was telling you about. Remember the wave, right? When the cell attaches and then detaches and pulls itself. So I went ahead and started stimulating the cell at very low concentrations of substances stimulus, right? Maybe these concentrations are the concentrations which cells communicate with each other. And what I saw is this. Okay. Is this working? Okay. So this is what I saw. Please notice how calcium moves inside the cell, right? Please notice the wave. So when it turns red, that means the signal is at its maximum, right? But look at the cell. The cell has a wave of calcium, right? It's not always red, right? It goes from the back of the cell to the front of the cell, and then it increases, right? So it's like a wave. Do you know how waves are? Do you know how waves are? They have oscillations, right? Then my next question was, if these cells are actually exhibiting waves of calcium, then why do we only see just why aren't we seeing calcium oscillations, right? So what I did is I went ahead and when I stimulated these cells with very small concentrations of stimulus, the same concentrations that cells use when they talk to each other, I went ahead and saw this, and then I was very excited, and I thought, can I measure the calcium? Do I have a substance, a spectrophotometer that will measure the calcium inside these cells while they are stimulated? And there was, right? Can I? Yeah. Okay, there we go. So as they were stimulated, and you see, there we go, these are the calcium waves. Interesting, isn't it? I knew there was something different. I knew it's just not that. Now the next step was to look at the poorly metastatic cells, the one that did not move much, right? That exhibited no movement. Are they exhibiting calcium oscillations? And the question was, and the answer was no, they didn't, right? So these were the highly metastatic cells. Do you see the calcium oscillations? And these were the poorly metastatic cells. No oscillations at all. The less poorly, the less metastatic but not extremely poorly exhibited 1 or 2 peaks, that's it. So there was a peak, but there were no oscillations, right? Then a crazy idea occurred to me, and I thought, you might think, why am I interested if the cells oscillate or don't, right? Why is it interesting to me that some cells oscillate with calcium and some cells don't, right? In my small mind at the time, I thought, maybe these oscillations are regulating the way the cells move, right? So every peak indicated a contraction and a pull of the cell, and every trough indicated an attachment, right? So more the calcium oscillating, the higher the movement or the faster the movement of the cells, right? I looked at different cells, and I looked at how they move and how fast they moved, and it turns out I was correct, right? So the highest metastatic cells, D145, 77.2% of them were oscillating, and they were oscillating regularly. And then the less metastatic, or the less migratory, 65 of them percent were moving, and then the 50, and then the lowest or non-metastatic, none of them were oscillating, right? So the next question was, first, how are they oscillating? Where's the calcium coming from? And next, if I were to stop these oscillations, or tune them in a way, can I stop the movement of the cells, right? Remember how we said these oscillations indicate cell movement? So if we stopped the oscillation, we prevented them, then maybe the cells would go ahead and be non-migratory, right? So first of all, we said, okay, maybe the calcium is coming in from outside because the hypothesis was it's coming through these store-operated calcium channels that I told you about. So what we did is we removed calcium from the outside and we stimulated the cells again, and when we did that, the oscillation stopped. So it started oscillating and then it stopped, which indicated that the calcium coming from outside is fueling these oscillations that are occurring, right? So if we were to stop the calcium intake to the cells from the outside, then the cells would probably stop. Yeah? And that's exactly what happened. So we stopped giving them calcium from outside, right? And what happened is, after these cells that closed in 16 hours, the wound that closed in 16 hours, after 30 hours, and even 48, we studied them further, it was still open. It didn't close, which means that the cells stopped moving, right? Very fascinating, isn't it? Yeah. Okay. When we gave them calcium back, the wound closed again. When I put calcium in the extracellular environment again, the wound closed. This means that these cells, these cancer cells that are moving so fast, that they're oscillating, right? They needed a supply of calcium from the outside in order for them to keep oscillating, in order for them to keep moving. When I stopped giving them calcium, they stopped moving, right? And they stopped oscillating. When I gave them calcium back, they went back ahead and they moved and they closed, right? This means that these are the statistics, we're not going to talk about it, which means that that channel I was interested in is probably doing something, right? Or it's probably overregulated or overexpressed at the surface. So we went ahead and we studied the mRNA expression, the genetic expression of these proteins. And these proteins turned out to be very highly upregulated in the cancer cells, but they were not present or very low in the poorly metastatic cells, right? This to me at the time was very interesting because, hey, I found two things. One, in order for the cells to move, they don't need just to exhibit a calcium increase, right? They need to be oscillating, right? Two, this calcium, in order for it to keep oscillating, comes from outside. And three, the channel in which it comes through is most likely ORI, the store-operated calcium entry channel, right? So to me, I found the target. And to scientists, when they find the target, that's like bingo, right? I got it. So my next step was, if I were to block that channel, right? If I were to remove it from the cell, would it be able to oscillate? Would it be able to migrate and move? Yes, you are right. The answer is no. So what I did is I knocked down, right, the channel. And this is what I saw. This is a normal cell that is oscillating. And this is a cell with 2% of the channel instead of 100%, right? And you can see the oscillations are no longer present, right? The next question is it still moving, right? So what happens is the invasion stopped. So this is, you can see the invasion and migration stopped. You can see in the pictures you see here. Not only that, but do I have cell migration? Yes, this is invasion. Okay, so the invasion stopped. So this is what you see in the highly metastatic cells, right? In the highly metastatic cells, you have the cells, the normal cells, and then when I took out the protein, what happened? There is no more invasion. And also, this is what happened in the other metastatic cells, where I put in the normal cells and then I took out the protein and the cells no longer invaded. The same thing happened with migration, right? For when I was talking about the wound closing, when I took out the protein, the wound stopped moving. It stopped closing, which indicated that the cells stopped moving, right? So my conclusion today, or my conclusion at the time, was two main things, right? As we all know, cancer cells, in order for them to proliferate, to migrate, to invade, they express calcium in very high amounts compared to those that poorly metastasize or move, right? The thing is, they don't just upregulate calcium. The calcium regulation in there is different, right? It goes in waves, and these waves are responsible for the way the cell moves, right? So the faster the cell moves, the higher the waves. The lower the cell moves, the lower the waves. So the question is, if we were to tweak these waves, so if we were to play with them, then maybe we can stop cancer cells from metastasizing, right? And this is very important because metastasis is the #1 killing cause of cancer, right? The next step was I found the target, which is the protein, OR. And OR, if we were to block it, we were to block these oscillations. And if we were to block these oscillations, we would block the cell movement, right? I'll leave you with this today. Thank you very much. Thank you for attending. Thank you. Any questions? Yes.
دكتورة انت حكيتي انه مرضى السكري بيعانوا من الانفصام انتروجينيسيس صح؟
Lack of angiogenesis.
وحكيتي انه هلا الكانسر بيحتاج انه يكون
Blood vessels.
الفيسلز تبعونه الخاصين فيه، فانت ممكن تحكي انه ممكن مرضى السكري بيكون اقل عرضة انهم يصابوا
Okay, this is a difficult question. There are no studies that show that this is the case, or no studies that I know of, right? This is a very smart question. This is a very valid question. بس الانجيوجينيسيس اللي بنحكي عنه هو الانجيوجينيسيس تبع الابيثيليال سيلز، مش بس الكانسر سيلز. الكانسر سيلز بحاجه بتبعت growth factors لحتى البلود vessels they grow further, right? What happens is that the cancer cells, they're always sending in signals, right, in order for the blood vessels to grow and metastasize. If that was the case in diabetic people, I'm not sure, but we need to look into it. Probably it will be fine. Yes.
How does this help in the treatment?
Which part?
Your conclusion.
My conclusion. How does it help in treatment? This is a very important question because what we can do. Remember that channel OI that I told you about? If we were to create drugs that would target that channel, stop it, block it, right? Then probably no more calcium is coming into the cell. And if no more calcium is coming into the cell, we wouldn't see these oscillations.
So there is nothing built on this.
Until now, no. This protein that I told you about is relatively new. People are still researching it. They're still looking into it. So it's all very novel. Yes. We would love to have that, بس للأسف ما عندناش التكنيكس. في عنا a lot of nanotechnological techniques that people are using. So hopefully the vehicle throughout which we target these cells is improved. لا مش بالضرورة، لأنه هي لما بتمشي في عندها borders، في عندها خط اللي هم البلود vessels والorgan tissues، right؟ رح تخبط فيهم مش رح تقدر تدخل unless it invades تتآكل السيل surface لحتى تقدر تدخل لجوا. That's another issue.
طيب بس ما فهمت ليش البكتيريا ما عندها ما بتقدر تعمل
poor regulation of calcium. They don't have enough calcium inside the cell اللي تخلي الvesicles to bind to the extracellular surface ويطلعوا Yes. Yes. Okay. Thank you. Okay. Yes.
So do you advise everyone not to take calcium supplements?
No, I'm sorry. So the calcium supplements that we're talking about is on a body level, right? It's on an organ level. The calcium that we're talking about is on a cellular level. I was talking about today was on a cellular level. And it's small micro controls of the calcium بالenvironment تبعت الcancer cell, right? It does not have to do with the whole body and organ level. بالعكس, I do not recommend against calcium. Calcium is very important. Yes.
The protein array.
Yes.
How did you stop? You said that
Yes.
for one.
Yes, there is a genetic technique called silent RNA knockdown, right? So what we do is we put inside the cells, I don't know what's your background, so I can go in details, but what we do is we go into the cell and we block the genetic expression تبعت هذا البروتين. So what happens لما يصير في عندي transcription of proteins لما يصير وقت expression أو وقت تصنيع البروتين it stops. It does not produce it. So it's not expressed. هلا we do not take it out completely عشان هيك لسه بنشوف شوي oscillation صح؟ ما عندناش technique to take it out completely ولكن ناخذ شوي صغيرة وهذا it decreases it completely. Yeah? They're telling me no more questions. مش عارفة.
No more
Yes.
question.
Yes, sorry. Thank you very much. Thank you. Yes.